Hello, this is Khadija. I'm back, and today we'll be discussing about ossification. So this is our outline. We're going to know what is ossification, types of ossification, the processes, bone remodeling, and clinical application. Although I think with clinical application applies with the bone remodeling. So definition and types. Ossification basically is the formation of bones and through from the mesenchymal stem cells through to the formation of cartilage and bone itself. So we have two types of ossification. We have the intramembranous ossification, which from the name is from the membranous type and it forms bone directly. And then we have the endochondral. Endo is inside, chondral means cartilage, so it is from a cartilage, okay, within a cartilage. So this diagram is basically showing you structures that develop from the intramembranous ossification. You can see the skull and the clavicle, and then the blue is structures are showing you things that develop via the endochondral ossification. You can see the long bone, you can see the vertebra, the pelvic girdle, and also the other long bones, which is tibia, ulna, radius, and the rest. So you can see the process starts from stem cells or mesenchymal stem cells that condense, and then you can see the arrow going to the membranous. So know that membranous also means intramembranous ossification. So you can see it forms the skull directly from the mesenchymal stem cells. So it doesn't actually need it to go through the formation of a cartilage. But in the endochondral ossification, you need to have a template formed by the cartilage, and then from the cartilage, gradually you have blood vessels coming in, and those blood vessels play a huge role in leading to the formation of the bone collar, and the bone collar induce hypertrophy of the cartilage. And then gradually this cartilage, you can see in number two, and then gradually in number 3 the cartilage is being replaced with green structure. You can see that is the bone and bony structure, but we're going to see this in detail step by step. So in this diagram, we want to understand what is the histology of compact and spongy bone, so that when we start talking about compact and spongy bone, you just don't get confused. So in this diagram, on the top left, you can see a bone, a long bone, and then you can see it has been cut open so that you can see the inner structure of it, including blood vessels. So on top there, you can see a blue cartilage on top of it and the bottom of it, and then you can see there is a separation between the upper spongy aspect. So the one you see with spaces is called the spongy bone, and the tough external of the bone is called the compact bone. So between the top spongy bone and the one below, you can see a thick compact layer. That is where you have the epiphyseal line. Okay, so note that is epiphyseal lines, but we're going to talk about the histology. So when you try to cut that off, like you can see it there, and you go underneath the microscope, you're going to see something like this. And from the inner aspect, you can see the spongy bone, which is in B, diagram B. You can see the spongy bone, and then on the lateral, on the outer surface, you can see round structure which are forming the compact bone. So the compact bone has 2 layers. It has an outer longitudinal layer, and then an inner circular layer. So these inner circular layers are formed by the osteoid, which are also formed by layers. So in diagram C, you can see it's just telling you just one of the osteoid bone has been removed, and you can see it's made of several layers layers, and inside you have the blood vessels in what is referred to as the central canal. On the bottom aspect in diagram D, you can see a magnified view of a spongy bone, which is actually similar to osteoid, but we're going to see that they are different. It's smaller than osteoid, but it's kind of similar but not the same. So this arrow is just showing you a magnified view of it, so you can see the osteoid. Every circular aspect, you can see blue arrow is the osteoid, is a cylindrical structure, and it has also layers surrounding it. So each layer is referred to as a lamina, and each layer of a lamina has the arranged circular arrangement of osteocytes. You can see it in yellow with the arrow is showing each cells as referred to as the osteocytes, and each osteocytes are connected by tiny capillary looking structure, which are referred to as the canaliculi. Those are important. You can see them diagonal and also transverse. So the canaliculi connects one osteoid to the next osteoid, and this is because when there's no blood vessels going deep down to the compact lamina layers, so the osteoid takes the canaliculi take blood and nutrient from the central canal, which has the blood vessels, and then they tap and transfer it to the osteocytes and other structures in that area. All right, so the parallel blood vessels are going in the central canal, while the ones on the longitudinal aspect are referred to as the perforating artery, and they are located in the Volkmann's canal. So this is now a magnified view of the spicules that makes up the spongy bone. So if you magnify the spicules, you can see also the spicules are also arranged in laminar form. They have the osteocytes, which are connected to the canaliculi as well. But in this, you can also see it doesn't have a central canal, but rather it is being supplied by if you see the perforating arteries go into the trabeculae, and on the surfaces of this individual spicules that forms the whole trabeculae, you're going to see the opening of the canaliculi. So if you go back to diagram D there and look at it, you're going to see the tiny opening on top of those trabeculae. So those are the opening of the canaliculi. So blood and nutrient diffuse through it, and it is able to send it down there. And also the external surface of the spicules or trabeculae, you can see a layer of cells. They are osteoblastic cells, and they are referred to as the endometrium. Sorry. So here is also a magnified view of the bone. So you can see the articular soft cartilage, which is the external surface. You can see the epiphyseal line, because this is an adult bone. So you have an epiphyseal line that has calcified. And then down there below, you can see the diaphysis. And so the proximal part and distal part are referred to as the epiphysis. The shaft of the bone is referred to as the diaphysis. And so between the two, you have the epiphyseal plate, and in adults they calcified, while in children and teenagers it hasn't undergone calcification, so it is referred to as the epiphyseal plate or the growth plate. So here you can see from the external surface to the internal, you have the diaphysis, you have the periosteum, okay, the external layer of bone, and it is a little bit cartilaginous and bony as well. So in that layer, you also have next to it, deep to it, you have the compact bone, and between the periosteum and the compact bone, there's a trap layer of osteoblast cells. Then deep to the compact bone, you have the spongy bone or the trabeculae, and then you have the bone marrow, which fills in the medullary cavity. All right, so here is just what we've discussed earlier on, and here in green now you can see this is an osteoid, and then we remove, you have the compact bone I told you is made of the external circumference lamina and then the internal osteoid layers. So you have several osteoid layers that makes up the inner compact layer. And then here we've seen a magnified view, and then you can see that there are several lamina around, and in each lamina you have osteoblasts, and osteoblasts are being connected by lines. Those tiny lines are referred to as the canaliculi. All right, so they are important. The osteoid is developing from osteoid deposition, and those osteocytes are the trapped osteoblastic cells, which we're going to discuss later in when we discuss the development. So you can see, don't forget we have two canals. One is one that we have the neurovascular bundles within the osteon, and then another one you can see the osteocyte. So this is the trabeculae. This is the central canal, and here you have the perforating canal, or you have it's called the Volkmann's canal. The next diagram is to explain the trabeculae, and you can see a magnified view of the trabeculae. Basically, from our previous slide, you can see that the spicules are open. The internal surface of the spicules, when you look at inside, it's similar to the osteon, but this one has no central canal, and it gets its blood supply via the openings of the canaliculi within the bone marrow. It also has osteoclasts ready to remodel it whenever it needs remodeling. And also you can see the trapped osteocytes and connecting canaliculi. So the processes, we said that we have the intramembranous and endochondral. So this in the blue lines, the blue cells are indicating that these develop from osteochondral progenitors, that is the mesenchyme and stem cells that can differentiate into osteocyte and chondrocyte. So here in the blue is telling us that that is when you want to do the endochondral ossification, and like the blue diagram. So we have the formation of a chondrocyte, and chondrocytes, when they align to give us a model, this model chondrocyte hypertrophy, that is they swell, and then gradually undergo degeneration and being replaced by osteoblasts. While in the intramembranous type of ossification, we say it doesn't have a model, cartilaginous model, so it just go from the osteoblast progenitors become osteoblast. They are being trapped as they secrete the osteoid, and then they become matured bones, and the osteoid calcified, and you have a stronger bone. So basically that is this as well. It's just telling us the differentiation of the mesenchymal stem cells like in A, and then this is basically A. You can see red become blue, green. Apoptotic chondrocyte means the chondrocytes are dead, and then you have pre-osteoblasts. Then that is the osteoblast. Then you have the matured osteoblast, which will become the osteocytes. And B is also describing intramembranous ossification. So you can see that A, endochondral ossification, has two layouts. It has the cartilaginous layout before it's being replaced by the bony layout. While in intramembranous ossification, you just have the mesenchymal stem cells becoming bone cells, and that is the osteoblast and the matured osteoblast, and then gradually become the osteocytes. And so C, D, and E is just telling us models that happen during remodeling structures, which are more basically clinical, and those people that want to go into molecular aspect. So we go deep into explaining intramembranous ossification, and you can see it's coming from the fibrous membrane inside, and then it occurs in flat bones. You can see it occur in the clavicle and part of the scapula, and as well as the cranium. So the first step of intramembranous ossification is the accumulation of mesenchymal stem cells are in a particular area, and these mesenchymal stem cells transform to become an osteoblast. And when they become an osteoblast, they start at an area referred to as the primary ossification center. And so this osteoblast starts to release a matrix substance which is called the osteoid, and this osteoid gradually starts to become more and more. So what happens is that as the osteoid is being produced, the osteoblast tends to differentiate, that is to multiply in numbers, and as they multiply in numbers, some of them in the periphery starts to open on the periphery, while the unlucky ones, the osteoblasts that are being trapped within, continues to be trapped, and they be transformed to become the osteocyte. So we go to diagram 2, and here you can see some of the cells trapped inside the osteoid are referred to as osteocytes, while the ones on the periphery continues to multiply and they become the osteoblasts. So now you can see how we start having the formation of a calcified matrix. Then by a diagram 3 is just telling us that blood vessels could come, and when blood vessels come in between to the inner aspect of the osteoid and the trapped osteocyte, it's bringing in new other nutrient. It's bringing in fibroblastic cells as well as stem cells, which could differentiate into osteoclasts. And these osteoclasts, what they do is they break down parts of the matrix, the osteoid matrix, and when they break down this part of this matrix to allow for the movement of the blood vessels to traverse that area. So when they do that, they form what is referred to as the spongy bone. Okay, that is what it is located in the medial aspect of the bone. While on the lateral aspect is that the osteoblast on the lateral aspect continues to deposit osteoid, and then some of the cells continue being trapped, and that is how they continue to form the layer of the compact bone. And when they have reached a particular aspect of it, they also produce a fibrous periosteum that lines the outside of the osteoblast. So you have on the outside you have the periosteum, and then you have trapped osteoblasts, then you have a compact bone, then you have in the middle you have the spongy bone formed by the spicules, and then inside you have a mesh opening for the position for the bone marrow. So basically this is how a flat bone is formed. All right, so now we go to the endochondral ossification. So endochondral ossification is a little bit different from what we just discussed from the intramembranous ossification, which is very easy. So starts from so mind you, ossification starts during the 6th week of development and continues all the way till puberty. And then you also have endochondral ossification also starts to develop also by the 6th week, but after 2 months the the cartilage models have started being replaced by the bone models, and this continues all the way till after puberty, when the epiphyseal plate becomes an epiphyseal line. So in this diagram you can see in the young person you can see the epiphyseal or growth plate, but by the time that person becomes an adult, you can see you have an epiphyseal line, and the bone marrow cavity is also reduced. You can see you have a stronger compact layer, and also you have the two epiphyses also calcified. So in this diagram is just showing a growing bone comparing with a matured bone, and so you could see how the position. So you can see the junction between the epiphysis and the diaphysis is referred to as the metaphysis, and this is important especially during the histology understanding of the growing bones. Alright, so here is just an internal, the diagram beside is just an internal structure showing what was happening between the growing bone and a matured bone. So above is just a growing bone. You can see a blood vessel within the epiphysis as well, and then you don't see that in the matured bone. All right, so that is to tell you that. And then you also don't see the cartilage of the epiphyseal plate, but now in the matured bone you see the epiphyseal line. And then look at the size of the articular cartilage in the growing bone versus that of the matured bone. So you can, those are things that you're supposed to understand. And also when you do an x-ray, these are things that you could actually observe, and you can compare. You can know if the person has actually is matured or not, because there are individuals that you don't understand age-wise, especially when you go into dwarfism and giantism. So these are things that help you identify that. So this is a little bit complex, like I said. So the steps of ossification, it starts from the 6th week and goes all the way till fertilization, till puberty. And then also it starts from the mesenchymal stem cells that differentiate into hyaline cartilage and forming the models. But after 2 weeks, those hyaline cartilage starts being replaced by a bony structure, and this bony structure at first of thing you get to see the formation of the bony collar, and this bony collar induces a lot of activities such that it prevents blood vessel easy diffusion of nutrient and blood to the inner cartilaginous structure on chondrocytes, and so they undergo expansion or swelling, and that's hypertrophy, and then gradually undergo apoptosis. And what happens is after, as they do that, we get to have blood vessels developing around that collar, and then it gradually goes into the developing bone. And when it does that, it brings in so many things, mineral nutrients, fibroblasts, mesenchymal stem cells. So those mesenchymal stem cells could differentiate into osteoclasts and osteoblasts. And the osteoclasts are very important because they actually eat off the remaining dead debris of the chondrocytes and giving us that internal spongy nature. And also the osteoblasts that are being produced could also lead to the development of other structures as well. And some researchers believe that some of the trapped chondrocytes undergo de-differentiation to give rise to osteoblastic cells, but that is still on the argumentative nature. All right, so then this continues to grow, the bone continues to grow at the epiphyseal plate due to proliferation of the chondrocytes. So basically these are the stages of endochondral ossification, but this diagram will explain more. So we have in A is telling us that is when the mesenchymal stem cells collect and condense. So when you hear condensation, they collect and proliferate more. They aggregate and proliferate at one point, and then they start to differentiate into and the inner cells start to differentiate into a chondroblast, and the chondroblast continues to proliferate, and then what happened is that there is going to be a deposition of perichondrial cells, and the perichondrial cells are important because they form the periphery or border for the chondrocytes. These perichondrial cells are important because they give rise to the foundation of the collarbone. So gradually the cells, the mesenchymal cells on the periphery will continues to align and give rise to the perichondrium proper around the whole cartilage, and and indeed there's going to be trapped mesenchymal stem cells. So you can see the diagram in C. So this trapped mesenchymal stems, you can see what is happening because they've started forming the collar around that area. They have reduced the diffusion of nutrient and blood into the deeper structure of the structure. So now you have the chondrocytes. In the middle, you can see they are swelling. That is in diagram C. They are swelling, and then and that's in the midline. Then as you go to the distal area, you can see proliferation. Some of them are resting, like the chondrocytes are resting. Then as you go to the most distal aspect, you see the proliferating chondrocytes. So from the middle, it starts to hypertrophy. They swell up, and when they swell up, because they don't have enough nutrient in them, and so they undergo apoptosis or death. And when they start to do that, and they start dying and creating space. So as that is happening, blood vessels starts to develop around the perichondrium, that is, if you can see in diagram C. So this blood vessels brings in with it fibroblasts and mesenchymal stem cells, which differentiate into osteoclasts and osteoblasts. So so some of the cells the osteoclastic cells starts to eat up the remaining hypertrophied chondrocytes. And don't forget that those chondrocytes originally have deposited the collagen and also some calcium and phosphate. So now what the chondrocytes, the eating cells, the chondrocytes starts to reshape the inner structure, while some of the chondrocytes de-differentiate to become the osteoblasts, and then also some of the osteoblasts are being trapped in the in the collar or the periosteum, or the so the like I said, some of the osteoblasts go into around the collar and they get trapped there, and as well they start to produce the osteoid. Then you find some of the osteoblasts being trapped, and so those that are being trapped become the osteocytes of the compact bone, while some of them remain in the collar bone area. So you can see in the central aspect is becoming a cavity-like structure. So the cavity-like structure is because of the osteoclast, which keeps eroding the middle aspect or the primary ossification center, and leading to the formation of the medullary cavity. And also osteoclast continues to reshape the deposited matrix, the collagen, as well as the osteoid deposited by both the chondrocytes and also the osteoblast, and which is going to give rise to the development of the spongy layer. And then when we go to E, so here you can see that the spongy layer has developed. You can see it is the most after the inner cavity. You can see the cells are trapped in the matrix, and then you can see the thick pink area that is referring to the compact bone. And then when you go on to the lateral aspect, you can see the periosteum. But before the periosteum, you can see trapped purple cells, which are the osteoblasts in this nature. So here you have the development of the diaphysis. So the bone continues to elongate, and they do that on the distal aspect, on the distal aspects as well. You can see it. This the chondroblast found on this the epiphyseal plate or the growth plate. They continue to elongate to increase the length of the bone, and also ossification will gradually occur in the secondary ossification area. Those are the ossification happening in the epiphysis, and this occur after birth, not before birth but after birth. So this is referred to as a secondary ossification center, which basically copies the same type of ossification that we just discussed, but this happens after birth. And when ossification continues to go towards the from the epiphyseal board to the epiphyseal plate, gradually what happens is that the cartilage undergo complete hypertrophy, and the epiphyseal plate becomes the epiphyseal line. So this is where you have the trapped perichondrial cells, and also the mesenchymal stem cells that really give rise to the formation of the perichondrium or the collarbone, and later on the collarbone. And here you can see where we have the trapped cells. So here you have from the lateral aspect, you have the periosteum, then you have trapped cells, trapped osteoblasts, then you have the compact bone proper, then you have the spongy bone from outside to inside. That is what you get to see. Then when you see the blood vessels, that is where you see the medullary cavity. So this is just showing you what we just saw right now. So you can see the bone collar forms around the diaphysis and of the hyaline cartilage, and then you can see that there is going to be deterioration in the central aspect of the cartilage matrix. That is where you have the primary ossification center, and that is where you start having the hypertrophy of the chondrocytes. And then gradually you have blood vessels coming in. When the blood vessels come in, they come in with mesenchymal stem cells and as well as fibroblastic cells. Mesenchymal stem cells differentiate into osteoblasts and osteoclasts. Osteoblasts give rise to osteocytes that deposit osteoid, while osteoclast continues to degrade and reshape to give rise to the sponginess. First of all, to give rise to the medullary cavity, and then give rise to the spongy bones, give rise to spicules, because the osteoclast eats around to give rise then to spicules. And then the lateral laying of osteoid and osteocytes it give rise to compact bone. And then at 5 you can see that at 4 you can see where you start having the secondary ossification center. This is what you see after birth. And then as from childhood to adolescent, you can see that the epiphyseal plate will gradually be narrowed, and gradually as ossification takes place, and gradually be calcified completely. So now this is what we've been talking about. Like you can see the primary ossification area in the first diagram, and then as you go upwards you get to see the hypertrophic chondrocytes, and then upwards you see the pre-hypertrophic chondrocyte cells. These are cells that have not, they have matured but have not gone under hypertrophy. And then you can see on top there you have the proliferating chondrocyte. So it's telling you that ossification starts at the primary ossification center and expands on both sides, so upwards and downwards. So as the ossification undergoes upwards and downwards direction, hypertrophy occur, and then you can see you have a resting chondrocyte because they are not doing anything. They just converted from a mesenchymal stem cells to a chondrocyte. They're just sleeping. But in this, it is in a child or a newborn child. And then you can see the articular cartilage, which is huge in this diagram. Diagram in the middle is just showing us the metaphysis. You remember where you have the junction between epiphysis and diaphysis. So you can see epiphysis here in the second in the diagram in the middle has shown that this is an area where the diaphysis has stopped, the diaphysis ossification have stopped, and but the arrow you can see from growth plate going downwards is showing you that this is the direction at which it's moving. So you gradually have apoptosis, hypertrophy, and hyper. So in this direction is opposite, is reverse to what we have in the long bone. So when you have ossification in the epiphysis, it is the reverse direction. So you are going to have hypertrophy from the middle, and then it goes downwards. And this diagram is also the same. It's just telling us this is where you have the resting, you have a proliferating chondrocyte, and these are just similar to the diagram. So this is just representing the same thing in the diagram. This diagram is talking about both primary ossification and secondary ossification. So in red, or the upper diagrams, is referring to primary ossification. So you can see how in the beginning, in the far left, you can see blue all the way. Then you can see green have started. It means that the mesenchymal stem cells, or the immatured chondrocytes, chondroblasts, have differentiated to become a hypertrophic chondrocyte, that is the swelling chondrocyte. And then gradually the chondrocytes, you can see in that growth plate, all the chondrocytes in the above the growth plates are remaining like that because they are resting. And then the ones on the midline, you can see the blood, they have hypertrophied, blood vessels have bronchiate, the mesenchymal stem cells, and also the fibroblast, and also the osteoclast and osteoblast, which tends to induce more hypertrophy to the osteocytes, to the chondrocytes, and then they gradually cell die. And you can see here in red, what is happening is that it's filled with bone marrow, and you have cells there. The cells are basically mostly chondrocytes and osteoblasts and osteoclastic cells. And then you can see the osteoclastic cells have given rise to the sponginess, and the second, you can see the secondary spongiosa. And then you have a green outer and red inner. Is telling you that the green outer has become calcified, is periosteum. The inner is now the endosteum, where you have the spongy bone covered with blood marrow. So secondary ossification is the diagram below. You can see the 1, 2, 3, 4 below. And so you can see secondary ossification center starts also from the midline and then spread gradually to the lateral aspect and also towards the epiphyseal plate. And then gradually everything calcified except the articular cartilage on both edges of the epiphysis. So this is also a reminder of the cell differentiation that we talked about earlier on. Okay, so here is also talking about the fossons that are found in the epiphyseal plate. Remember, the most cranial of all is the resting phase. And then the second phase, you have the proliferating phase. The third is the hypertrophy phase. And the fourth is the calcified cartilage, or that area where you have the plate forming. All right, so basically that's how the cells look like, but they gradually undergo ossification from the midpoint, and then gradually go radiate outwards towards other sides. So this is also describing this diagram, the lateral diagram, and you can see how ossification starts to happen. And in the epiphysis you can see secondary ossification center, and then gradually radiate outside. So it starts in the reserved zone and then spread towards the other directions as well. So this is just a summary of what we've discussed so far, and you can see how in E and F is telling us how from cartilage the cartilage ratio between E and F. So you can see F is almost an adult. You still have a plate there, but by the time the plate with more years, the plate is going to completely calcify and becomes a line. So clinical application, you have bone growth and also bone remodeling. So this is bone growth, means the increase in length, or the increase in the length of the bone. So this is linear growth. You have width growth as well of the bone, but linear growth is mostly by the cartilage, while width growth is mostly by the position of the osteoid by the osteoblast cells. So linear growth of the bone is also stimulated by sex hormones, which eventually inactivate the epiphyseal plates. That is when big plates becomes line. So you can see what happened is that at the epiphyseal plate or the metaphysis, you still have the proliferation of chondrocytes, and that was the arrangement zone, zone 1 to 4 you saw earlier on. And then you can see how the junction is it's located. You can see from the diagram below, you can see the thickness in that area. You have dividing, the one more distal is the dividing, and then you also from the bottom aspect closer to the plate, you have the hypertrophied cartilage. So bone remodeling could be during a trauma or deficiency of vitamin A, or you have low bone density or osteoarthritis, which could, if not corrected, leads to osteoarthritis, or during bone fracture, you need a bone remodeling, which is a physiological aspect of our body. Our body does it physiologically. So bone remodeling process starts maybe when you have a resting phase, and then you have injury, a trauma, any kind of trauma, and then what happened is that it takes like 3 weeks for it to have the osteoclast remove the dead cells, and then you have also, because of blood vessels that are going to accumulate there, they are going to remove the macrophages, and after that osteoblasts is being brought as well by as mesenchymal stem cells by the blood, will start the cells transform into osteoblasts, and then gradually they become they start to deposit osteoid, and some of the cells become trapped and they are referred to as new bones. So this would take about 3 months to do that. So here is the stages of bone healing when you have a fracture. So what happened is that the first stage is called the hematoma, where blood accumulates in that area. It does a lot of things leading to swelling, triggering the immune response. That is white blood cells also come in to remove all bacteria or infection in that area. And also it brings in mesenchymal stem cells that would convert themselves to one fixed and the blood vessels, the ruptured blood vessels, to also transform themselves to be the relevant type of cells we need, like the osteoblast and osteoclast cells. So next, this there is going to be the formation of a fibrocartilage callus that will form there because of the deposition of the chondroblast cells. And then when they form, they do give rise to the model. It's going to be replaced by a bone-like structure because of the osteoblast cells. And when they do that, they're going to give rise to an irregular shape, and so we need osteoclast to come and remodel or reshape the area, like straighten it up, remove the excess bony callus to give rise to a normal bone formation. So these are the stages of bone healing or remodeling. Thank you very much. I hope you if you have any question, please let me know, share it in the Slack group or WhatsApp me. Have a good day.